You're on mute, Kate.
Fred, I'm glad you're in here.
Hey, nice to see you again.
Good to see you. Awesome.
I made it, but there might be some dogs barking in the background.
I was supposed to be in my doctor's appointment 30 minutes ago, and they still haven't seen me. There's two people in front of me. So I'm going to have to like pause myself at some point and leave the conversation. So I'm glad there's some folks here that can keep it running.
No problem. I should be able to stay for the whole session, so shouldn't be a problem.
Excellent. Yeah, very good. Okay, good deal. And Doug should be here as well. And I don't know if I know the USGS folks, it's a federal holiday, so they didn't get permission to work, I guess.
Oh,
Okay.
well, I know I think on the McGill and the ISRC side, I think I'll be the only one, but
That's okay. Yeah. But your perspective is fantastic.
did you have, I didn't see, I'm not really prepared actually for anything, but what was the idea, is just to have a discussion about it, or?
Yep, that's it. It's
Okay, perfect.
It's going to be pretty relaxed, I think.
Perfect.
Yeah. I think the plan is for PSG to have more, like present our data and really talk about the event specifically on Middleton, but today we can talk even in more generalities. Just because we've got all sorts of other potential participants with this one.
Perfect.
And Jeff, thank you so much for helping today.
Problem.
I don't know if you heard me, but I'm waiting in a doctor's office right now. My appointment was 30 minutes ago, and there's still two people in front of me. So at some point, they're probably going to call my name, maybe. So I'll definitely, I'm happy to kind of admit people or whatever. If you're admitting people into the session, that's great.
Yeah, don't worry about that. I'll admit people, and should make sure people can share any presentations if we're doing that, and anything else.
I don't think we have presentations. I asked Doug Causey, since I'm talking on my cell phone, if he could share just a figure that I had put together. So at some point he'll probably need to share his screen. But I don't know. I mean, we can keep it pretty fluid because there's not really any talks during the panel that are planned.
I still have the really quick presentation that we had on our meeting last time with the videos of the die-off. If we need, I can just find it quickly on my computer.
Yeah,
so
if you want to do that, just to do like the intro to what we're kind of what we have dealt with, or what happened this summer at least.
Yeah, perfect. I'll just pull it up quickly, and if we want to give a little just background information on the die-off, that'll be it.
Yeah, that's great.
Okay, Fred, nice to see you again, by the way, but I made you co-host.
Perfect, thank you. Nice to see you again too.
Awesome. We'll probably just give it a couple more. Yeah, Ricardo, I'm on my cell phone. Sorry, you're just gonna have to deal with my mug.
if you don't mind, I'm just gonna try to share my screen, just make sure it works well. That's
Yeah,
okay.
and if you, we'll just give it like 2 or 3 minutes, Fred, and then I'll kind of get the ball rolling and I'll let you.
Perfect. I'm just gonna try it quickly and then stop sharing, just to make sure it's working so that,
Wait.
can you guys see that?
Yes.
Perfect. And I'll stop sharing and I'll keep it open for later.
We're just gonna give it a couple more minutes, folks, and then we'll kind of get going. I apologize. I'm on my cell phone, sitting at a doctor's office. So I'm 40 minutes late for my appointment, or they're 40 minutes late for my appointment. So I'm stuck here, and we'll be just transmitting from Pennsylvania today. All right, Doug, I'm glad you're here. Awesome. Okay, I think we can probably, we've got most of our participants, at least from the summer die-off. Hopefully everybody can hear me okay. My name is Kate Sheehan. I'm at Frostburg State University. I am an assistant professor there. And over the summer, we had a very interesting seabird die-off in Middleton Island, in the Gulf of Alaska, where it appears that there was an avian botulism outbreak, and it resulted in 100 to 200 birds dying. And we had a really interesting opportunity with these because they were on, yeah, my phone's gonna move, guys, sorry. I can turn off my video if that makes you more comfortable. So we had this particular die-off, and we had the opportunity to preserve specimen with that. And there were some wonderful field researchers that were out on the island. So there was an opportunity to get birds that were preserved in good condition shortly after dying, and even kind of monitoring them as they were on their way down. And then kind of a interesting call went out because there was an unknown and we weren't exactly sure what was going on. So this opened the door for some collaborative opportunities where we had different federal agencies involved, different universities involved, and other individuals. So that kind of sparked this well, I guess it was just kind of what happened. We had this kind of collaborative effort that went down in order to get the most out of these birds. And kind of the lessons learned and the progress of that collaborative effort it is still unfolding at this point. But we wanted to have a conversation because these outbreaks don't seem to be getting any less frequent. We are seeing increased reports, and then of course, as we're dealing with zoonotic diseases and things like that, the risks to human health are something that we're also wanting to monitor, or maybe now it's being recognized that it's more important to monitor these things, that we're being very careful when we are dealing with samples, especially of wild animals. So we kind of put together this this session, and we're doing this at PSG as well, although we're going to be presenting some more specific results from the Middleton die-off itself. But what I wanted to do today was to kind of open a dialogue in terms of what it is that we're seeing in the field, whether or not the animals that we see that are kind of dying potentially from infectious diseases or something that can be sampled for assessments out in the field. And then what happens to those samples, those specimen, things like that? How can we make the most of them? Because again, it seems like these occurrences aren't getting any less frequent. In fact, they're getting more frequent. So Fred Tremblay is here, and she's one of the researchers that was on the ground in Middleton, and she's got a short presentation to give everybody kind of a background in terms of what happened this year in 2021. So we'll kind of kick it off with that, and then hopefully, if I'm not in my appointment, I will be able to, we'll kind of have a broader discussion, not only about this event, but about other events and potentially future possibilities with this. So with that, I'm going to mute myself and let Fred talk.
So, hi everyone. So I'm just gonna quickly share my screen. So as Kate mentioned, I'll really be speaking from the field perspective. So I'm a master's student at McGill University, and I also happen to be directing the field station this summer on Middleton Island. And so what I'm gonna review here is really the perspective we had from the field of this die-off from the start to the end as it was occurring. And so just to give you a little bit of background. So Middleton Island is this tiny island in the Gulf of Alaska. And there are two main black-legged kittiwakes colony on the island. One that we call the main colony, where kittiwakes have reclaimed essentially old Cold War buildings, and one that is on an old shipwreck. And so just a little bit background. So the kittiwakes there are nesting on artificial buildings because the cliffs have gone way too eroded. So it's a bit of an unconventional colony for that. And then there's also glaucous winged gulls that are scattered all across the island, mainly ground nesters. And so we've also been able to monitor glaucous winged gulls through this period. And we've monitored the glaucous winged gull mainly at the North Lagoon that you see in green, where there's a bit of a higher density and a lot of vagrant as well as nesting individuals. So the black-legged kittiwake colony around the buildings, this is what it looks like. This is just essentially to give you an idea of where they nest. And all of these names there, so the Blicky Tower, the Skelton Tower, the West Towers, these are all the buildings essentially that we monitored on a daily basis, actually twice a day for every day during the whole die-off. And then this is what the ship colony looks like. So this one is below the cliffs by the ocean. And there's also, along with the black-legged kittiwakes, there's also tufted puffins, common murres, pelagic cormorants, and glaucous winged gulls that are nesting in the area. And so this is just a quick timeline of how the die-off started and what happened. So we first noticed an odd number of kittiwakes dead around the main colony on July 16th. And to give you an idea, we usually see maybe 10 dead kittiwakes in our whole season, which is usually 4 to 5 months long. And in this one day, we noticed 10 dead kittiwakes and then started noticing 32 individuals acting sick. And so not being able to fly or having a lot of trouble taking off. And then quickly after the start of the die-off, we started also seeing glaucous winged gulls dead either on the side of the road and showing similar signs of sickness than the kittiwakes. And the glaucus winged gulls was especially alarming because they nest in much lower density. And so the fact that we were noticing the glaucus winged gulls as well was quite unusual. And so by July 18th, we'd reached what we estimate is the peak of the die-off, where we had only 3 dead kittiwakes that day, but over 34 sick kittiwakes, and that was only around the main colony, not including the ship colony. And so this is what kind of like the peak in sick and dead birds looked like. And so we really peaked around the 18th and then slowly started dying to reach the end of the die-off that we approximated around the 27th of July. So here, every dead individual is a newly dead individual, as we collected individuals that were dead on a daily basis on both the main colony and the ship colony. And so these carcasses that we collected were then shipped off the island, and then we slowly stopped detecting carcasses until we stopped monitoring as we were getting off the island on August 13th. To give you an idea of what those behaviors look like, so the first video is kind of like the early stages where the individuals are still mobile, but aren't really able to fly. So you see these individuals trying to flap, but not really being able to take off, but still being able to run and get away from us a little bit, especially the second individual. Then if I stop this one, we move on to what we call the next stage. And so what we see here is this individual still has the capacity of flapping its wings a little bit, but cannot stand up anymore, stumbles really easily, but still has some capacity to move. And then the later stage where only the head essentially was mobile. And so usually we'd see kind of that process happening in about the first 2 or 3 days. They'd be somewhat mobile, and from the day that so when we see the third video, essentially when they reach that stage, they would usually die within 1 to maybe 3 days. Oops. Sorry. And so just to kind of wrap this up quickly, so during the whole length of the die-off, we collected really a broad range of samples, including 73 carcasses from kittiwakes, glaucous winged gulls, and even 2 herring gulls. We collected over 36 carcasses from black-legged kittiwakes. We sampled with swabs 28 birds for avian influenza, as this was the first cause we investigated, and then collected regurgitation fecal samples from all species present on the island, not only the black-legged kittiwakes, collected blood samples, also blue mussels, and did essentially a daily monitoring of both the kittiwake main colony and the ship colony, as well as the lagoon and other areas on the island, and essentially made a detailed diary of all the events. And then finally, just acknowledge I'm speaking here today, but I wasn't alone. I had an amazing team of technicians, researchers, professors, and so all of this sampling would not have been possible without them. And so this is just essentially the quick overview of the die-off on the island.
That's great, Fred. Thanks so much. There are a couple of questions, it looks like, in the chat, and I just got pulled into my doctor's appointment. So if you want to answer some questions, that's great. And if not, then Doug, if you want to introduce yourself after Fred's done answering questions, and anybody else that was part of the Middleton that's on the panel, if you want to introduce yourselves, you are welcome to do so, but I'm going to mute myself for a little bit.
So there's a question in the chat that Doug is partially answering right now from Ian. He says, have you considered paralytic shellfish poisoning, such as sexy toxins? There have been kills of Arctic terns attributed to PSP in southeastern Alaska in recent years, and a recent paper in PNAS reports fields Alexandrium cyst in the Chucky and Beaufort Sea. And so that's a really good question. So our first impressions was that it could have been actually a potential harmful algal blooms, so including saxitoxins or domoic acids. And that's partially why we started collecting blue mussels. So the way we investigated this dia from the field perspective is starting with what could be the worst possible scenario. In this case, we were thinking about highly potent avian influenza, and then moved on to the kind of like least worrisome opportunity. But saxitoxins was definitely one of the first things we started looking into. Doug, I don't know if you want to introduce yourself or if anybody else has more questions.
Well, hello everyone. I'm Doug Causey. I'm here in Anchorage. I'm at the University of Alaska Anchorage, how I first met Fred and the rest of the team was an enormous shipment of dead birds, I might say that what's somewhat unprecedented about this is not that there is die-offs of birds, but Fred and her team were there, made observations as you saw, and then this is almost unheard of, was able to freeze and stabilize the specimens. Usually that's not the case. So with that, we were able to undertake all of these subsequent examinations. When you do this kind of work, like Fred said, you start with, you make a list of what the potential causes are and start with the worst and go down the list essentially. Highly pathogenic avian influenza is certainly known for seabirds, and two of the graduate students in my lab are looking exactly at that. And as most people know, what is extremely concerning about that is that it's highly pathogenic. It's a variant of influenza that humans can get. So that was the first thing we tested. None showed any positives, and we did a sequence of tests from extremely detailed survey of the actual presence or not of influenza RNA. Did not find that. We did find a few individuals with what's called avian influenza, and most birds carry influenza to no impact. And but the presence and the type of that influenza were expected, so that clearly was not an issue. And Ian asked a question about biotoxins associated with harmful algal blooms, which we're starting to see now in the Bering Sea, Gulf of Alaska, and Aleutian Islands. And we focused on two of the best known and most lethal, actually, of the biotoxins, which is domoic acid and saxitoxin, and didn't find that either. So the testing that we did of the specimens to determine what pathogens could be possibly associated with this, and it clearly was a pathogenic effect. That allowed us to use the same data and go a little bit further into the what were the sequences that we discovered. And primary was the DNA associated with Clostridium, which is what causes botulism. And from that, specimens were analyzed both here at the University of Alaska Anchorage as well as the National Wildlife Disease Lab, and it was determined for several of the specimens. I should tell you, we have not looked at all of them. That there was the presence in the specimens of botulism C, which is a form commonly as well, almost exclusively associated with birds. And good news, it is not a toxin that affects humans. So on small evidence, but it's positive evidence nonetheless, where we are right now is to assume that the birds on Middleton Island had been infected with Clostridium, which is normally found in fresh water. So these are marine birds on an island in the middle of the Gulf of Alaska. So where the heck did this come from? And one obvious source would be the freshwater ponds on Middleton Island. But Clostridium shows up also in intertidal organisms and some other sources. So this work is very much going on right now. And Kate, as I see you're back, what Kate and I did was when we got this large shipment of specimens, we determined a necropsy panel. And Kate, I'll let you talk about the parasites that you looked at.
Sure. So, and I'll mention one thing about the potential mechanism for spread of botulism C is that it can run through larval dipterans. So maggots of biting flies
Yes.
are known to transmit. What they'll do is they will create meiosis infections. So you have the maggots that are essentially deposited by the adults. The maggots, the eggs, right, hatch, and the maggots live off of tissues of living organisms. So you've got these birds that are basically having these maggots being laid in their orifices, we'll say. And we've gone through a handful of, we've gone through all of the gastrointestinal tracts that were not given to the USGS for harmful algal bloom assessments. And we found maggots in the majority of them, whether they were in the esophagus or in the colon or in the cloaca, really. So it appears that there was this strong potential for that botulism to be spread in this instance by insects. And presumably those insect populations are maintained to some degree with those freshwater habitats that are on this island. So yeah, I think those freshwater ponds are really playing a important role here in the maintenance of botulism on Middleton Island. But I mean, it's still very preliminary in terms of the data that we have as far as the parasites are concerned. We've found really high species richness of parasites in the glaucous winged gulls so far. Black-legged kittiwakes, pretty similar parasite richnesses and similar types of parasites to what we were finding in wild, presumably healthy birds that we had collected, Doug and I had collected in 2016 from the Western Aleutians. Sample sizes are not super high, only like a dozen or so kittiwakes to compare them from our previous work. Glaucous winged gulls, I only got 2 GI tracts to look through. USGS got the other one to look for harmful algal bloom stuff. But it doesn't seem like these guys, it seems like their declines were fast enough that it wasn't necessarily impactful on their parasite communities. The parasites that were there, you know, they didn't lose anything along the way as they were starving it appears. Yeah, so that's kind of what I have to report on the parasite front, at least. I guess I was interested in, you know, like, so this is a really interesting, and, you know, we had this necropsy party. Doug, you talked about the necropsy party a little bit.
How are you?
Good. I need 3 minutes. I'm really sorry.
Yeah, I can take over, Kate. Okay. Kate went mute. Sorry.
Sorry, Doug, I'm here for a second.
Okay.
My dog just walked in the room. We coordinated kind of a training session, a necropsy party, to try and make sure that we could get through all the birds, or the majority of the birds that we had at the University of Alaska Anchorage on campus, and kind of had a training session, right, to make sure that everybody was doing their necropsies in the same way. And really, I was interested in trying to make sure that we could be measuring the same things, and doing it in a very standardized, methodical way, so that we can compare. And if we do this in the future, that we can standardize with other assessments and other potential outbreaks and those sorts of things as well. So we got together and we processed 30 birds in 4 days, between myself, students and researchers at UAA, USGS. We all got together and made that happen, which was really great. And I think by the end of it, we learned a lot, and we were all pretty much on the same page, which is great. But there were still some instances where we were having to make decisions in terms of who's getting what sorts of samples. We were doing harmful algal or HAB assessments. USGS needed like a minimum weight for a particular organ, or they needed a particular volume of digestate and those sorts of things. So, you know, looking at how large a kidney was helped us to determine whether they were going to be able to run their tests on that particular organ. And if they couldn't, they would give it to me for parasite assessments, or if they could, or if we cut it in half, right, we could split it and divvy things up in that way. So I think, you know, again, it was a really great opportunity for us to understand if we could share samples, and if we could get more information out of an individual bird, as opposed to, you get this bird, you process it for what you want to do, we're going to get this bird and process it for what we want to do. Because, you know, it seems like we should be able to get multiple assays out of the same individual birds, as opposed to moving birds around. So I kind of wanted to maybe open up like a general discussion. Doug, you know, I'd love to hear, you know, you and I chop in your 2 cents on this as well. But I think, you know, I feel like this is the kind of direction we should be trying to move in, to have collaborators, you know, figuring out how we can get lots and lots of data out of each individual bird, as opposed to needing lots of birds to get lots of information about a population that's involved in a die-off. So I'm gonna shut up and listen now.
Well, if I could jump in here, I might underscore again why this was of first such interest in the opportunity that we have. This is the first recorded mass die-off of birds associated with botulism here in Alaska. Now, I emphasized recorded because, as you all know, Alaska is really isolated, and particularly where the seabirds are nesting and breeding. But whether it's the first or the second, it still is very unusual for this to occur. And mass die-offs caused by botulism do occur in the lower 48, excuse me, that's the rest of the US, and has been associated with the blooms of Clostridium. Botulism is botulism A is a listed toxin. Okay, that's how dangerous it is, and that would be botulinum type A and B, which affects humans. The other types are for birds and other mammals. So the concern here first is to understand what caused this, and there's birds have been breeding on Middleton Island for a long time, and the freshwater ponds have been there a long time. Why did it happen now? And again, what's unusual about Middleton Island as a site is that it's one of the few places in Alaska that has been continuously monitored for decades. So this is not an incident just because by random chance Fred and the team were there, but this is the first time it has ever been recorded at one of the most studied seabird sites. So some of the conjectures that have been made is that because of the general warming temperatures in the Gulf of Alaska and Bering Sea, and this includes not only the air temperature or land temperature, but the oceanography, that somehow this is related to a bloom of Clostridium. We unfortunately don't have that information. The second is that obviously from a conservation and management standpoint, we need to understand more about what are the correlates between a deadly outbreak like this. And a question that is very commonly asked, in relation to these types of wildlife diseases, is that it was very episodic. It did not continue the entire breeding season. As Fred showed you in her great presentation, there was a small period of time when you had a mass die-off, and then there was really no mortality caused after that. So the approach that we're taking first is to try and understand what was the cause. And then this is where we would compare work that's been done in other breeding areas in the lower 48. See if we can come up with a possible connection with, is this a random event? Is it something that we can predict will happen in the future? Also, like a lot of wildlife pathogens, that not every individual dies, and so here the interest is, are there any subsequent effects? And one of the aspects that Kate was looking at is that how does the Clostridium, the botulinum associated with Clostridium, does this have any effect on the micro and macrobiota in the bird itself? So like Kate said, we did this initial analysis on 30 of the specimens. We have another roughly 40 as well, that we are going to be doing similar kinds of analysis to get a better idea of what is the incidence, what are some of the downstream effects of a lethal exposure to botulinum. And finally, that this gives us a lot of information to be able to start looking at what will the future lie. For example, next year, the great concern here is not, one of the great concerns here beyond the conservation and management isn't so much Middleton Island. This is a place that's awfully hard to get to, but these are species that are commonly used for subsistence by indigenous communities throughout coastal Alaska. And botulism, like I said, type C generally does not cause human illness, but there's so much that we don't know. So there is a lot of reasons that we want to continue this research. All right, so that's pretty much all I wanted to say at this moment. Any questions, Fred? Do you want to add to this?
No, I think from my perspective, this was very much of a first time exposure to die-offs and toxins, parasitology. And so I'm here more as of interest to see where the discussion essentially is going to lead everyone. And so, yeah.
Okay. Well, now I'm going to be like a professor and call on people to answer, but I won't do that. Does anyone on the call here have experience with events like this? Jenna, I see you just turned yourself on.
I don't have any experience with this, but just out of curiosity, I know you were saying that you don't know what the cause of it is yet, but do you have any reason to expect that this could occur next year?
Well, Jenna, that's a really good question.
I don't know how botulism works, so I don't know if it can if it could persist over a winter or.
Well, it's a toxin that comes out of a blue-green algae called Clostridium, and in one sense, it'd be like any freshwater algae. When the conditions are good, it has a bloom. Well, why did it stop so quickly, and why didn't it happen before? I mean, okay. Good question. We have no answers.
Yeah, it seems so odd to me because this didn't seem like a particularly odd weather year in any way. And we have had odd weather years in the past few decades. So yeah.
Yeah, so if I can add to this, this was kind of what was going on in our head too when botulism kind of started showing up in the results, because you've been there in 2019, weather was super warm, which seems to be conditions that are much more likely for botulism. But one pattern that we did notice is that the die-off started quickly after a heat wave on the coast. And so we didn't see it. We didn't feel it on Middleton. For us, it'd been pretty foggy and not that warm, but it was following the big heat wave on the western side of the coast. And then the die-off lasted probably something like 3 days. And we stopped seeing new cases shortly after a large rain event. And so this is, don't take this for a fact or anything. This is just observations that we made, but it seems like this rain event somewhat essentially stopped the die-off. And so.
Jeff, if you're able to make me a co-host, then I can share my screen. Okay, good.
Your code.
Thank you. All right. Okay, now I have to find. Now I have to find it. I think this is no, that's not it. There we go. All right. So are you able to see this figure? Somebody? Yes. Okay. So this is a chart of the GPS foraging tracks during this period, and at the bottom where it says start, that's where Middleton Island is. And then the where I'm moving my cursor here, this is on the entrance to Prince William Sound. So here, and then I'm going to move to another one. That is for chick-rearing black-legged kittiwakes, and then here is the GPS tracks for the black-legged kittiwakes during the die-off. And although the Jenna, again, this is why your question is so great. I mean, it seems like the freshwater ponds on the island would be an obvious source of the botulism, but these are seabirds that forage, okay, and so they very likely could have picked this up at another site on this island right here, or in some of the other foraging areas. Dave has a comment. If you look at the chat, that botulism E is responsible for mass die-offs as well, but in the northern tier of the U.S. and southern tier of Canada. So again, what we have is intriguing data, but making the connection we're not able to at the moment. And I'm going to stop the share now. That's why we're doing these very detailed necropsies, as well as I think there's a plan to head back to Middleton Island next breeding season. Fred, isn't that right?
I'm sorry, you cut out slightly, so you'll have to repeat.
Isn't there a plan to head back to Middleton Island next year?
Yeah, so usually every year there is a team. I may not be there, but there's going to be a team of monitoring there for sure. And so we're definitely staying tuned for any signs of this die-off reoccurring next year.
Okay, so let me see. Dave has provided, I mean, in chat, some related papers on this from the events in the Great Lake. And the question from Jeff is, what are the population sizes of the black-legged kittiwake in comparison to how many died? Fred, that's up to you.
So I'm currently looking to find our census number. I don't remember by heart how many individuals we counted this year, but we are in the 10,000s, I believe, right now of kittiwakes, and our glaucous-winged gull census was 9,000 and something, if I remember properly. And we counted, we observed 300 and estimate that there's at least double that actually died. And so we're talking about maybe 600, 700 individuals out of a population of 10,000 kittiwakes.
There's a question by Kayoko. Is there a possibility that the large amount of droppings of birds cause the eutrophication of pond and the bloom of algae? Well, yeah, absolutely, but this happens every year. Fred.
Well, just to add to this, I know we showed the map earlier where they foraged. We see the kittiwakes use the ponds regularly, but we don't see them forage. I've never ever observed the kittiwakes there foraging in the ponds. And so it might have been in contact through bathing or something if it is coming from the local ponds, but they don't forage there. And so that's making it a little bit less likely. But at the same time, there are some ponds that have changed over the years and that made it different this year than other years. I'm thinking of one pond specifically where we used to have a dam that kept the water level quite high and used to be a frequently used spot for bathing for the kittiwakes. That following the breakage of the dam last winter, the water level dropped quite significantly. And so this is one of the potential sources that we think, okay, maybe this change in water level coupled with the high amount of droppings in this water has made this pond maybe a little bit more specifically prone to develop. Sorry about that. Sorry, I have a dog with a broken leg, which is unhappy. But yes, so it could come from a freshwater source, but it's a bit unlikely.
All right.
Forget if you sorry, I forget if you mentioned, but were any of the alcids or cormorants impacted? I think I remember seeing somewhere that there were maybe a few, but not many. And if go ahead.
That's a really good question. No, actually, we monitored every single colony, including the puffins, the rhinoceros chocolates, the cormorants, really closely to see if we would see any signs. And we didn't see any. There is maybe one potential case of a murre being affected. I didn't observe it personally, but it was unclear whether this murre was just essentially unable to take off because it was on the ground, or if it might have been affected and we weren't able to get close enough to this murre to actually know for sure. And so my conservative answer would be no, it was really restricted to kittiwakes, glaucous winged gulls, and a few herring gulls.
Again, Jenna, that's a really good question, because here we're looking at how does the natural history, the ecology of the species affect the potential acquisition of the biotoxin. And as most of you know, we're talking about seabirds that feed right in the intertidal. Some are, as you can see, like I put up, the black-legged kittiwakes are moving across a rather large expanse of the Gulf of Alaska. Some are feeding near shore, some offshore. Okay, so that these are ecological correlates, I guess, of which birds were affected and which are not. And I'll tell you, like Fred said, my experience has been black-legged kittiwakes do not use freshwater ponds and for bathing either. So to get, oh yeah, Kate, are you back now? Kate. Okay. Well,
yes, I am. Sorry, I was looking through the chat.
Okay. That's why we have the detailed behavioral observations that Fred and the team were able to do. Our detailed necropsy is allowing us to, again, and the necropsy allows us to not only look at the potential macro and micro and macro biota, but we can also look at the effects on organs and tissues. And so this gives us a really detailed potential for a really detailed understanding of what is associated with this rapid die-off, and how we compare this to, well, excuse me, what we would compare this to is work like Fred mentioned, Kate mentioned, and we've been doing elsewhere in the Bering Sea, is that we can compare what we find in these birds that died with birds that are healthy and are without any obvious impact of something like this biotoxin. All right. Well, I think everybody knows who I am. But if you have any here, I'll put my email address in the chat. If you have any questions or want to follow up with any other observations, please contact us. Again, we are using this as being able to give us insights into, is this going to be a consequence of warming, the warming environment, whether it's freshwater ponds, the land itself, or the oceanography. What aspects of what happened tie in with what's being observed, for example, in the Great Lakes, and what impacts does this have for conservation and management?
I have a question. Is there a plan for the beginning of the season for Middleton's next season to do more thorough surveys to try to catch the beginning of this again, if it does happen again, or?
Well, Jenna, I'll ask that quickly because it's easy for me to say I don't do any of the work on Middleton Island. Okay, that'd be up to the Fish and Wildlife Service and USGS, but it would make obvious sense to do.
Yeah, I guess I was wondering if Fred knew who was going to be taking over as crew lead next year and had been in contact with coming up with a plan for what that might look like.
I cannot speak as of the new crew leader for next year, but I know that Shannon has now taken over the Institute and is working really more tightly with Scott, and they are both very aware of the issue and of the risk. They couldn't be here today, but they are both very aware that this might be a recurring issue and are starting to take the steps and thinking too as ways to mitigate if this is a recurring event and how to deal with this in the future. And so it's a work in progress.
Cool, thanks.
Well, and something maybe worth adding and maybe talking about too, that so like field kits, Fred was really lucky to be on this island with freezers and lots of garbage bags apparently, and all these things that allowed for them to monitor and really get a hold of these things and do a great job in preserving them for us in short order. But when it comes down to it, a lot of the times the places that we're out in the field, right, we don't necessarily have those conditions where, and part of it, maybe discussing what it is, how we outfit Middleton Island in general to help support collections of animals if and when this happens again there, or if samples, cloacal swabs, to make sure we have kits and something that's slightly standardized and maybe a checklist, something like that. So it's not just, oh, this person needs this sample and this person, right? To actually have a really streamlined way of being able to do things so that it doesn't seem, it is chaotic, but it may be a less chaotic when it comes to that. And, I guess that's something, again, that it's not as hard to do with Middleton because they are connected, literally. But I guess I'm wondering, maybe from the audience, the participants and things like that, if those sorts of kits might be helpful in terms of development, and if we had some sort of centralized way to get those out to the field to folks who are willing to maybe collect samples if you're finding dead birds, potentially diseased birds on your field sites. Has anybody got anything to say about that?
I don't have anything to say other than I think that's a great idea. I know I work at the Alaska Sea Life Center, and I know when we heard what was happening on Middleton, we definitely were trying to be even more vigilant. And I think it would be great to know, who to contact or how to get a kit like that if we did start seeing things, so we can start doing whatever collections or whatever was necessary from the beginning.
I see a comment from Ian on the shellfish poisoning. Yeah, so I think these are the sorts of things that are absolutely worth, so who would test for it? Right, which is wonderful. So how do we collect those samples? How much tissue do we need? What sort of tissue do we need? Those are the sorts of that's the sort of information that we need to have to figure out if we want to be able to do these investigations and not necessarily only rely on the U.S. Fish and Wildlife Lab to get to some of these answers. If we want to share samples and share samples from individual birds, we've got to also kind of come up with a list of who does what sort of thing too. Because we know we have some individuals that are really interested, at least in Alaskan populations with harmful algal blooms. They may not be as interested in processing birds that die off in the Gulf of Mexico. I'm not really sure, right? So also like trying to figure out who are, like who would be really interested in taking on samples if they were collected from a particular die-off. I think that's also really important to just identify, and then again, making sure that we're all in alignment with what sort of methods they need, or what sort of samples, how big they have to be, that sort of thing, so that we can appropriately divvy things up to get the most information out of each bird.
I might add that the conclusion that these die-offs were not directly caused by harmful algal blooms has a lot to do with what's called the reportable limit, and this is similar to almost any kind of a disease or epidemiology setting where concentrations or presence below a certain amount is not considered to be associated with any kind of an effect. And we are, however, continuing to study and test all of the tissues that we have using not only our lab but some local labs that here in Alaska that are skilled in doing this. And so that's what I was saying earlier is that the levels we detected were not high enough to be able to make that conclusion. But as you all know, when we collect data, we don't, we're not so much interested in does it meet the level or not, but what is the actual data. So stay tuned. Well, if there's no other questions, I put my email address in the chat. Like I said, I'd be surprised if anyone doesn't know how to get a hold of me. Just Google my name, and I unfortunately show up really easily. But we are planning to put our results, not only the 30 birds that we examined as a basis for the statement that was released, but we will continue the work on the rest of the samples that the field team was able to send to us. All right, everyone. I don't have any more. Kate?
I guess I have one last question for Kate. If you did come up with one of these like kits or something like that, how could people connect to learn about that?
Yeah, thanks, Jenna. So we're going to be doing these kind of sessions, workshop panels. If you're going to PSG, and a bunch of us are going to be talking next week as well at the Alaska Bird Conference too, so we can connect there as well. But this is something that we're kind of developing at this point, right? So we're trying to kind of have this conversation over and over again with a handful of different audiences, and I think this was our first attempt at it, and that's awesome. And I'm really glad we've had so many people in attendance and interested in what happened here. And I think probably, maybe the next iteration of this, we can either come up with you know, like a workshop or something like that, to where we can actually develop these things and put out feelers in terms of what actually needs to be in those kits, because I think that would be a fairly easy thing to put together. It's then just disseminating those and having exactly, you know, what you're bringing up is how do we get 50 of them to stockpile to keep out in the field, or to put those in their field kits kind of things. So it's something that we're putting forward, and it's TBD right now. So we'll help us. I'd love to have you participate and figure out what would need to be in them, so that we can then kind of get them out to field crews wherever they might be, so that we can standardize some sort of collection technique and share samples as much as possible. Awesome. All right, do we have any other questions? We've got like 9 minutes left if we want to use them, or I don't see any new messages in the chat. Sorry, my fingers on the moving over my screen here.
Okay.
Awesome. All right. Well, thank you so much for all attending. And if you want to learn more about this, we're going to have kind of the official talks, maybe with some more official results presented at PSG. So that's another opportunity to hear more about this.
And,
Go ahead, Doug.
okay, and then also for those of you who are in the area, we're also going to present at AMSS, that's the Alaska Marine Science Symposium. So stay tuned. This is very much a continuing story.
All right. Thank you so much for participating, guys. Appreciate it. And we'll see you in another session.